All right, guys, we are on to chapter 11. This is interactions between microbes and humans. So this is in general, how do we interact with our microbes, both our home team and also pathogens? So when we talk about our home team, we're talking about our normal human microbiome. So this is all of our normal biota, which there's a handful of different names. So our home team, human microbiome, normal biota, resident biota, aka our normal flora. There's tons of names to describe those normal microbes that live in and on our entire body. So there really is just a gigantic balancing act between our home team and pathogens because our microbiome is going to be one of our first defenses against pathogens from causing an infection in our body. When it comes to our microbiome, it's not just going to be bacteria. It also includes fungi, protozoans, viruses, arthropods, as well as some of those little buggies, those parasites, right? Those helminths. So it's not just bacteria that is included in our microbiome. So make sure you know that. Now, when it comes to the relationships that we have with all of our resident biota, that's where the Human Microbiome Project, or HMP, comes in. Now, we've talked about this guy before. This is a big research project, a big co-op, essentially, that takes samples from all kinds of people, all kinds of places, and all kinds of samples, literally from everything from spit to hair samples, skin samples, samples from your eye, your nose, your mouth, other orifices, as well as other liquids or solids that you can think of. Literally, the imagination, it doesn't stop there, right? So the HMP is going to look at all kinds of relationships that we have with our normal biota and see what happens with our microbes that live in and on us. What do they do for us? What do we do for them? That leads us to what has the Human Microbiome Project found? A few things that are kind of interesting have a lot to do with our diet and how we are able to digest certain things, how we can break certain products down, or more accurately, how we can't break certain things down without the help of our home team microbiome. So things like milk, as well as being able to make certain vitamins, thanks to E. coli, things like that. Super important relationship. Another thing that the HMP has found is that initially we thought that quite a few areas of our body were sterile, and it turns out that they actually aren't. An example is our eyes. Initially, it was thought that the eyes, the surface of the eyes, were sterile environments. However, what we have found through the HMP is that the eyes have just as much bacteria, and specifically the same species of bacteria as our skin, which is kind of cool. So that leads us to microbes that are not part of our home team. So those microbes that are pathogens that can make us sick. Now, just because we come into contact with a microbe that is a pathogen, that doesn't mean we're going to get sick. We need to talk about a couple of terms like infection. This just means the growth of a microbe. It does not mean that you are deviating from being healthy. Okay. Now, a lot of people think that the term infection means diseased. That does not mean the same thing. Again, infection just means that the microbe is growing. When it comes to the term disease, however, this is specifically a departure from health, where the growth of the microbe is causing injury to the host. There are two types of disease that we need to talk about: non-infectious disease and infectious disease. Now, let's talk about those. A non-infectious disease is not directly caused by microbes. An example are some cancers, heart disease, alcoholism, diabetes. An example that has been changed is ulcers. Initially, that was thought to be a non-infectious disease, but now we know that that is actually caused with a relationship with microbes. So again, non-infectious means that it is not caused by microbes, whereas an infectious disease is caused by a microorganism, like the flu or mumps or chickenpox or gonorrhea or the plague or tetanus. Now, when it comes to different types of infectious diseases, there are two categories we need to talk about: communicable and non-communicable. Remember, those are types of infectious diseases. So, a communicable disease is a disease that will spread from one host to another. An example being chickenpox. If you can imagine it, back in the day, they used to have chickenpox parties where one kid would get chickenpox and then all the moms would get on the phone and be like, hey, little Susie has chickenpox. Do you want to bring over Billy and Tommy and Caitlin and bring everybody over so all the kids can get chickenpox together so that they essentially all have chickenpox all at once, right? Now, chickenpox is highly communicable. When it comes to non-communicable diseases, these are diseases that are not spread from person to person. If you get that disease, if you go lick somebody else or cough in their face, they won't get that disease. An example being tetanus. The way you get tetanus is kind of an old wives' tale, is you step on a rusty nail, right? Or you cut yourself on a rusty tin can lid or something like that. Now, it's not the rust that causes the disease of tetanus. It's actually the puncture of that metal, because tetanus, the microbe, is found in dirt. And it's the dirt on the metal being punctured, injected into your body, that is the actual mode of transmission. Now, again, if you have tetanus and you go up to somebody and you start smooching on them or spitting on them or coughing on them, they will not get tetanus because it's a non-communicable disease. However, if you have chickenpox or the flu and you start kissing somebody, spitting on them, coughing on them, whatever, they are very likely, if maybe they don't have the vaccine or an immunity to that disease, they will likely get chickenpox because it's a very communicable disease. So infectious diseases, there are three kind of parts that have to sort of align in order for you to get sick. So some important things to keep in mind is that not all contact is going to lead to the colonization of that microbe inside of your body. So just because you come into contact with chickenpox, maybe in passing, does not mean that you're going to get it. And not all colonization leads to infection. So let's say you come into contact with chickenpox and you get the chickenpox microbe into your system. Does not mean that it's going to lead to that infection, because it might not take hold. And then not all infections lead to a disease itself. So not all microbes are going to be pathogens. That's just kind of the way it is. So most of the time, you won't know you could have gotten sick. There are so many times in a day, in a week, in a month, in a year in your life that you don't know that you came into contact with a microbe, that microbe potentially didn't colonize in your body, or that it did and it didn't cause a disease. So there's just a lot that has to align in order for you to actually get sick and show symptoms. All right, some more terminology, some types of diseases. Now, these two terms are often used synonymously, but I need you to understand that they do not mean the same thing, but a lot of people think they do. That is communicable and contagious. Now, we've been talking about this word communicable. That means that a disease can spread from one host to another, right? Now, it means it could spread. Contagious means that a disease is highly communicable, means it has a higher likelihood that it will spread through contact. All right. A good example of a highly communicable disease is measles, which is a pretty good thing to think about right now because measles are coming back up. Measles are highly communicable. And one of the reasons why measles are kind of so scary is that you can spread measles without showing signs that you are sick. So that is why it is so contagious as well, is you're walking around acting like you're fine because you don't know you're sick yet, and it's highly contagious. Meanwhile, something like leprosy is very weakly communicable, right? So it's not contagious, it is weakly communicable. This is a condition that causes lesions on the outside of the body. There used to be, back in the day, leprosy colonies because it was thought that leprosy was highly contagious, but it is not. You'd have to essentially like suck on somebody's wound in order to catch leprosy. Now that we know that it is weakly communicable, but back in the day, hygiene wasn't as big of a concern. So that is probably likely why leprosy was transmitted so much. And we are moseying right into non-communicable diseases. Again, these are the types of diseases where you're not going to be contagious. You are not going to be able to give somebody else this disease if you kiss them or spit on them or cough on them, right? This is stuck inside of you. And in fact, I want you to think of it that way. The microbe itself doesn't want to be in you. You are not the intended host as far as humans go, right? So we are not the microbe's goal. But once it gets into us, this microbe can't get out. This is why we are called a dead end host for these non-communicable diseases and microbes. So the end all result of a non-communicable infection is that we get sick because the microbe's going to die because it can't go anywhere. It's kind of like in the death throes. It wants to get out. It can't, so it's going to die. And in that process of it can't get out, it's going to die, it starts acting up in a way where it's going to take us with it when it dies, and it's gonna really throw a tantrum on its way out. So there are essentially two options, because again, no matter what, the microbe is going to die inside of us. So again, like tetanus. We get tetanus from the environment, and the result of tetanus is it causes paralysis in our muscles. Now, tetanus is going to die no matter what. It's just going to be, are we going with it or not? Those are the two options. Either we die or we survive this non-communicable infection. So let's take a look at our home team just a little bit closer. Starting with our skin and eyes, because there's a lot of overlapping microbes between these two areas. So When it comes to the skin and the eyes, both are kind of hostile environments. The skin is not a great environment because it's dry and salty and acidic, and the eyes aren't great because it's exposure to light, as well as our tears having a lot of enzymes. So, not a great place in either way for microbes to exist. It's pretty rough. Now, when it comes to the skin, there are some positive results when it comes to our home team, and that is that the presence of Staphylococcus epidermidis, which is on everybody, and it covers all of our skin and also up into our hair, super fun, um, is that it creates essentially a layer of competition with other microbes. So that is a positive result. Same for our eyes. Essentially, you have S. epidermidis also in your eyes as well. The negative result, however, of our home team is that we have minor skin infections. I want you to think of like a maybe a small cut, like a paper cut on your finger, and around that cut, you might see a little bit of inflammation, like a little bit of redness and puffiness around that cut. That is an infection that is just surrounding the cut, right? Of our home team, most likely just causing a small infection surrounding that cut. It's just acting up. Body odor is another issue. I want you to think of your groin and your pits. Those are areas that are going to smell particularly unpleasant if you don't shower or bathe regularly with soap. That bacteria is going to go through metabolic processes in consuming lipids from your sweat. And if you do not bathe properly and frequently, then our home team gets a little bit stinky. As well as acne can also be a negative result, having to do with getting into our pores, blocking up our pores, and making fun little pustules and whatnot. Another fabulous place for our microbiota is, of course, the mouth. And you've probably heard, things like, oh, the human mouth is the most disgusting place, blah, 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 blah. Yeah, no, it totally is. It's a really disgusting area. This is why you should be brushing your teeth every morning and every night. Because it's an ideal area for microbe growth. It's warm, it's wet, and it's full of nutrients, as well as a lot of oxygen is going in and out, as well as carbon dioxide, because some microbes like carbon dioxide as well. So there are all kinds of fabulous microbes that like to live inside of your mouth. Keep that in mind. Isn't that fun? The positive result, much like our skin and eyes, is that we have protection from pathogens in the sense that our home team is out competing the pathogens that get into our mouth because they're just taking up space, right? The negative results are things like cavities, bad breath, plaque, which plaque is an example of a biofilm that builds up on our teeth. And you can see from that bottom picture that this person's teeth, that yellowing around the edges of the bottom of the teeth, that is plaque. If left for long enough untreated by a dentist, that plaque is going to eat away at the enamel of our teeth, as well as just eat into our gums as well. Super not a good time. So those are some negative results that can happen from our home team if we do not treat our home team properly, as well as our mouth properly. Make sure you brush your teeth, guys. Very important. Going pretty hand in hand with the mouth is going to be the specifically upper respiratory tract. This is where we have a lot of our normal microbiota as well. I want you to make sure you know that this is the upper respiratory tract and not the lower. The lower respiratory tract should be sterile. We don't want biota growing in that particular area. If we do, it's like it's pneumonia or bronchitis, something like that. Not a good time. However, In the upper respiratory tract, which is the nose and throat, you have oodles of microbes because it's a fabulous place to grow. Once again, kind of the same area of why is warm, moist, full of nutrients, and also lots of airflow and carbon dioxide. So think about all of your snot and mucus. All of that is fabulous areas for microbes to live within. And of course they do. So, positive result of our microbiota living in our upper respiratory tract is going to be protection from pathogens. You're probably going to see a theme here that is the main primary positive result of our microbiota. However, the negative result of our microbes living in the upper respiratory tract is they're very, very close to the lower respiratory tract and often cause a few issues if they fall down into our lungs. Again, pneumonia, bronchitis, that is not a fun time. So that is the negative results of our normal home team causing issues. Moving on to our next, essentially we're going system by system. I hope you can kind of tell that. Moving into our next system of where we find our normal microbiota is the gastrointestinal tract. And we're going to break that down into two areas, the stomach and the intestines. And I know your anatomy teachers are probably going to come for me in just clumping the intestines into one. I know there's the large and the small intestines, and we both know that. We're just going to clump them together, guys. We have the stomach, which has a very important feature, and that is stomach acid, which has a very low pH of 2, meaning your stomach acid is so strong it can eat through metal. This means that for the vast majority of microbes, they don't get past our stomach. That stomach acid is going to kill most microbes, aside from some extremophiles that are going to live there. For example, Helicobacter pylori is going to live in the stomach lining, and if it acts up, it can give you stomach ulcers. So we've been talking about stomach ulcers a few times here. That is the microbe that causes some issues if it acts up. That's part of our home team. In our intestines, we have all kinds of microbes. Now keep in mind that stomach acid stays in our stomach. In the intestines, that pH is very different because of what is released from the liver and the pancreas and whatnot. So in the intestines, we have a lot of diversity of species, some 400 different microbe species. A very important one being E. coli. E. coli is going to help in synthesizing vitamins like vitamin K. Very, very important. We have symbiotic relationships with the microbes in our gut. They perform things like vitamin synthesis, as well as help us digest cellulose, things like that. Super important. Very, very important. Some negative results from the microbes in our intestines is that we can get things like diarrhea if those microbes act up, as well as gas production. So if you're feeling a little bloated, a little gassy sometimes, it's usually your home team responding negatively to something you've consumed. All right, here's our last system where our home team is going to reside, and that is the urogenital system. Now, everybody's got a urethra. The difference is going to be kind of how long the urethra is in males and females. And that is going to be kind of the difference in how certain infections can manifest when we talk about kind of that relationship of what can go wrong, right? So in males, due to the presence of the penis, urethras are longer, meaning that the end of the urethra is going to be further from the anus, as opposed to females, where the urethra is particularly short, and the location is going to be particularly close to the reproductive system as well as the anus. So the likelihood of getting an infection is much higher for females than it is for males. So that is one of our negative kind of things to note here. Now, when it comes to the reproductive system in general, that is also another difference between males and females. The presence of the vagina is the biggest difference, of course, what we're talking about here. For females, the vagina is going to be a warm, moist, but kind of acidic environment, a little bit on the hateful side, as she should be. This is going to prevent and limit microbial growth. That is the hope and the goal of the vagina. We don't want things to grow in there. And in fact, one of the evolutionary purposes of menstruation to bleed and slough off the inner lining of the uterus is so that pathogens, if they do get up into the uterus for some reason, do slough off and are shed monthly. So kind of an interesting little side note there. Now, when things go wrong because of the location of the anus, which have, and again, we've already talked about this, 400 potentially different species of microbes, that is so close to the vagina and urethra in females, there is a very high likelihood of getting UTIs, as well as infections of the vagina, because of the proximity to other systems, right? So that is different for males versus females. Now we passed over some systems because that is not where we want to find any microbes. Those are sterile environments. Things like the brain, your blood, your kidneys, your liver, those should be completely sterile. And if we have microbes growing in those areas, there's a good chance that maybe there was a puncture or a wound. Something has gone horribly, terribly wrong to where there are microbes in those sterile environments. And we potentially have a bigger problem than just there's microbes there. Potentially there is something else. Now, the lungs and the bladder were initially thought to have, to also be sterile environments. However, in recent times, that Human Microbiome Project has come to light that those areas might have just a handful of microbes. We are still, for the purpose of this class, going to count the lungs and bladder as being sterile environments within the body. However, guys, it may be important to keep an eye on this later in life through your career, that maybe that changes. Now, typically, anywhere where we interact with microbes on the outside of our body or wherever there is an orifice, that is how we acquire our normal flora as far as those normal microbiota. So think of your skin, your eyes, your mouth, urethra, any openings that are exposed to the outside. So that makes sense that that is going to be that area where we see that interaction happening and thus that acquiring of our normal flora. These sterile areas, however, should not be exposed to the outside. For example, your brain, my God, it should never be exposed to the outside. And if it is, well, again, we've got bigger problems if it is. Some microbiota might be present on our bodies or in our bodies for a short period of time. We call these transient microbiota. They won't stay with us forever, maybe a few days or weeks or months, but usually they'll take off after a bit. They're kind of just passing through, if you will. But our normal microbiota are going to be permanent because we have a symbiotic relationship. I've used this term a few times already, but we have symbiosis. This is a relationship or a partnership with our biota. So let's talk about these different kinds of partnerships that we can have. Symbiosis can be mutualistic, commensalistic, or parasitic. Mutualism is going to be beneficial for everybody. So both partners in the relationship have a beneficial experience in the relationship itself. For example, the bacteria found in our intestines are able to eat and have a space to exist, as well as they help us synthesize vitamins as well as digest food. So we benefit, they benefit. Mutual benefiting, if you want to think of it that way. Commensalism is where one organism is going to benefit while the other organism is unaffected. In this case, the microbe benefits, but the host is not affected. We're not harmed, nor are we benefiting. And I don't have a great example for this one, just know that it can happen. And then parasitic is going to be a bad relationship for us. Now, the microbe benefits. However, the host is harmed. An example is like pathogens, just in general, like influenza or tuberculosis. These are going to be microbes that cause disease. So I've said it a few times, but we're going to give it a term now. Our home team is constantly competing with pathogens. And the cool thing about it is our normal microbiota is it has the advantage of having the home field advantage, if you want to think of it that way. So they have the space, they have the resources, they're already established, they have the numbers. And when our home team is competing with those pathogens and usually wins, what we call this is microbial antagonism. So our microbes are antagonizing the pathogens. And that's a super cool thing. This is going to be one of our first lines of defense against pathogens, is our own home team essentially out competing pathogens from establishing themselves inside of us. So our home team, as well as just our own immune system in general, work together in tandem to make sure that we don't get an infection. Now, what happens if we do? What makes us more susceptible to getting an infection? And there's a laundry list of different things that can cause individuals to be more susceptible to infections. Number one, age, and that's being either older or younger. So these are individuals whose immune systems, and again, that includes our own immune system, think like white blood cells, or our home team, which is still part of our immune system, to not function as well or to not be set up quite as well yet. So another thing that could happen is maybe you're fresh out of surgery or you had an organ transplant and your system is just kind of frazzled or stressed out. Another thing can be stress. So physical or mental stress, that's a big one, as well as something like pregnancy, where all of your resources are being put into building a new person. So speaking of fetuses and newborns, let's talk about when we develop our home team. Typically, this is going to be when we break fetal membranes and expose the infant to microbes. This is usually during birth. So let's talk about it. So babies are usually born one of two ways, and that is vaginal birth or through a C-section. So either way, the baby is going to be exposed to all kinds of microbes. So let's talk about vaginal birth first. On the way out, the baby is going to be touching all sides of the mother's reproductive system, so the vagina, right? And on the way out, the baby's going to be touching mom's skin as well as all kinds of surfaces. Once the baby is out, typically the first thing that we do in the healthcare field is we put the baby onto mom's chest. And usually the goal is for the baby to be touching mom skin to skin. That serves a handful of purposes. One, fantastic feeling for mom of relief that they are actually holding their child. Two, that is going to start the baby's immune system, their microbiota. All right. And where they're getting their microbiota is directly from mom. So her skin microbes will now become baby's skin microbes. Now, the same thing is essentially going to be happening with the process of a C-section. However, it's just not that process of a vaginal birth of touching every side of the vagina, but we are still going to probably put baby onto mom's chest to get some skin-to-skin contact. Same with dad. You're probably going to be getting a lot of touching from mom, from dad, grandma, grandpa, all kinds of people. And this is going to immediately kickstart that normal home team for the baby. Because while in the womb, it's a sterile environment. Baby's never seen a microbe. So we got to get that kick-started pretty quick. That moves us into breastfeeding versus bottle feeding. Much like a vaginal versus C-section, there's not a whole lot of difference actually between the two processes. A vaginal birth is going to get the baby's skin microbiota to register faster than a C-section, while with bottle feeding versus breastfeeding, the only difference is we are establishing the gut bacteria faster. Specifically, this particular bacteria that I want you to know, which is called Bifidobacterium. We cannot replicate this in formula. However, it is important to feed a baby no matter what. So bottle-fed infants are getting little to no difference other than this single bacteria, which the only thing it does is it establishes the gut bacteria just a little bit faster than in bottle-fed infants. Again, little to no difference between vaginal versus C-section babies, little to no difference in bottle-fed versus breastfed infants. Now, for newborn infants, it is very important to expose them to all kinds of things right off the bat. So in the first 24 to 48 hours after birth, microbes that they come into contact with will become the home team. After that point, it's sort of like a door shuts and that's it. No longer accepting any more applicants. So once that has ended, the gut bacteria, the skin bacteria, all kinds of bacteria, that's it. So it's important to establish that home team in the first couple of hours after birth, because once that has ended, then again, we're not accepting any more microbes. Now let's talk about a relationship that is not a good one, that parasitic relationship with microbes, a pathogen. So this is when things go wrong. So when we talk about pathogens, we need to talk about a couple terms and a couple things, like how strong a pathogen can be and those different factors, what kind of disease that a pathogen can cause, and then like the effects that will happen, how strong those effects will be, etc., etc. So let's jump into it. We have this term pathogenicity. This is going to be the potential for a microbe to cause a disease. And pathogenicity is going to be going to divide pathogenic microbes into two groups. One, true or primary pathogens, and then the second is going to be opportunistic pathogens. So let's talk about these two groups in a little bit more depth. So what's important about true or primary pathogens is that these are pathogens that are going to be capable of causing healthy individuals with a normal immune system to become diseased. This is very important because that's going to be the difference between true pathogens and opportunistic. So true pathogens are going to be pathogens that make healthy people sick. That is the big thing here. All right, opportunistic pathogens, contrary to those primary pathogens, those true pathogens, will do one of two things. Cause individuals who already have a compromised immune system sick, or they will move from an area that they normally don't cause a problem in to an area that they do cause a problem in. So let's talk about it. That first one, in individuals who already have a compromised immune system, I want you to think of somebody who has like HIV. They have a virus that makes them already immune compromised. A regular pathogen or an infection of any kind would not have to do very much in order to cause an infection and a disease in someone who has HIV because their immune system is already down. For that second part, the or, right? When the pathogen moves from an area that it normally would be fine in, maybe like E. coli in the digestive system, that's perfectly fine. We love E. coli to be in our digestive system. If E. coli moves from our intestines to, say, our urinary tract, that causes an infection, a UTI, urinary tract infection. That is not okay. So that can be a result of essentially like location, location, location. That can be a big issue. So when it comes to opportunistic pathogens, these guys won't make everybody sick. It just depends on what's happening. If it's a pathogen that infects somebody who's already immune compromised, or it's a pathogen moving to an area that is not prepared for that pathogen, like E. coli moving from the digestive tract to the urinary tract, those are the two kind of big options here for opportunistic pathogens. So that leads us to this term virulence. Virulence is how severe the disease is going to be, and usually we phrase it as the degree of pathogenicity, which again, pathogenicity is going to be the potential for the disease. And a lot of times virulence and pathogenicity are used interchangeably, but do keep in mind they don't mean the same thing because we're using this term pathogenicity to describe virulence. The virulence of a microbe is going to be determined by two things, whether or not the pathogen can establish itself in the host, and if once it's established, can it cause damage. So we're going to talk about these as virulence factors. So these are characteristics or structures of the microbe that contribute to the preceding activities of the microbe itself within the host. Now, we have these two terms, infectious dose and lethal dose. This is going to be an amount of cells to perform some function, and the function itself is going to make sense based on the term. So the first one is an infectious dose. So how many cells to make an organism sick. So sometimes called the ID50, and it has to do with how much of the microbe do we need to make a notable infection in 50% of animals that we're testing, essentially. It is the ID50, the infectious dose. However, I care more that you know that it takes X amount of cells to make an individual sick. The next is the lethal dose. And I'm sure you can kind of put this kind of together, that if the infectious dose is how many cells to make an organism sick, we need to talk about how many cells it takes to kill an organism. Now, that is the LD50 compared to the ID50, so infectious dose versus lethal dose. And we do want these numbers to be relatively far apart, because an infection is fine. We can fight an infection. However, that lethal dose can be the scary one, right? We want those numbers to be pretty far apart, and the closer the numbers, the shorter the time between the initial infection and then death. And that's where infections can get particularly scary if they cause death. All right, last but not least, when it comes to this first part of Chapter 11 is polymicrobial infections. Since the beginning of infectious disease studies, we've essentially had this concept of isolating single microbes. This relates back to Koch's postulates, and if you want to look back at that, go back a few chapters and take a peek at that. We're going to look at that a little bit later in this chapter again too as a review. But essentially, we've had this concept that this microbe causes this disease, and we use Koch's postulates to help understand and focus in on that concept. However, when it comes to the majority of infections, we now understand that most of them are polymicrobial, meaning that there's more than one microbe involved in causing that infection. An example being influenza and pneumonia, that influenza is the starting infection and will frequently lead to pneumonia. It kind of almost opens the door for that infection to come in, but they will overlap simultaneously. Poly meaning multiple, so we're saying we have multiple infections here. So that is the end of this first part of Chapter 11. Join me for this last part. It's going to be a pretty hefty section for this last part. So happy studying, and if you have any questions, let me know, and I'll see you for part 2.